Okay, do we have signal all around? Okay, we're rolling. So what we're going to do today is we're going to finish that story I started yesterday with the electrocardiogram. And I want to emphasize when talking about the electrocardiogram that there's only one or two ions that we're talking about. We're talking about sodium influx and potassium efflux. And all of those movements of ions are occurring in the muscle cells, whether it's the atrial muscle cells or whether it's ventricular muscle cells. That's what we're measuring. And we'll walk through what that means. And what I'll try to do as we walk through the electrocardiograms, I want to bring you around to the idea of how that will fit with the pressure volume curve and try to put that whole story together. And then we're going to move on. We're going to talk about autonomic nervous system and regulation of the heart. And we're going to see the autonomic nervous system be broken down into these two great groups. One group is said to be the sympathetic branch. The other group is said to be the parasympathetic branch. Typically, when you hear this term sympathetic, you think fight or flight. When you think the term parasympathetic, you think rest and digest. And we'll walk our way through this, particularly in view of the heart. Now, if you take another neurobiology course, take another physiology course, you may get much more deeply into the autonomic nervous system. And you'll find the autonomics controlling all manner of things throughout your body. But we're going to use the heart as sort of the quintessential organ that's going to be innervated by the autonomic nervous system. We're going to walk our way through that system. And we're going to see that it's a good place to think about it because the heart itself is autorhythmic. And yet we can modulate, we can change the rate and rhythm of the heart through the autonomic nervous system, and we'll walk our way through how that gets done. To understand this, we have to first talk about autonomic anatomy. Now I'm going to walk through this in two steps. The first step is going to be to talk about just the autonomic anatomy. We're not going to get down into the distinction between the sympathetic and parasympathetic just yet. And there's some commonalities of these systems that I want you to hold on to. And the commonalities are that whether we're looking at the sympathetic branch or the parasympathetic branch, the preganglionic neuronal cell body is always in the central nervous system. We'll see in some cases in the spinal cord, some cases in the brainstem, it's always in the central nervous system. That preganglionic neuronal cell body, whose cell body resides in the central nervous system, will send a process out into the periphery. It sends a process out into the periphery, and the preganglionic neuron synapses onto a postganglionic neuron in autonomic ganglia in the peripheral nervous system. Now, if you extend that logic, you'll see that the autonomic ganglia are located in the peripheral nervous system. So we're going to see the preganglionic neuron always synapses onto the postganglionic neuron. The preganglionic neuron has its cell body in the central nervous system. The postganglionic neuron has its cell body in the peripheral nervous system. And the synapse between the pre- and postganglionic neurons will occur in these ganglia. Then finally, the third step is the postganglionic neuron is going to innervate the target. So two-cell chain, preganglionic, postganglionic. The target can be one of three things. The one we're going to talk about here today is cardiac muscle. But there's also a possibility that it could innervate smooth muscle, and we'll talk about that a little bit tomorrow when we talk about the smooth muscle of the vascular system. Or it could innervate a gland, and we'll talk about that when we get to something like the digestive system. And we'll talk about the parasympathetic innervation of the, say, the salivary glands. But hold on to the idea that we have two-cell chain, preganglionic, postganglionic. Preganglionic cell body CNS, postganglionic neuron is in the PNS, and it forms synapses on the target in the periphery. There's some pharmacology we have to go through because it's the pharmacology that lies at the heart of how this system functions. And the pharmacology is pretty straightforward. Whenever you see the pre to postganglionic synapse, it doesn't matter if it's the sympathetic branch or the parasympathetic branch, whichever branch of the autonomic nervous system we're talking about, the preganglionic to postganglionic synapse is mediated by acetylcholine. The receptor that mediates the acetylcholine response is a nicotinic acetylcholine receptor. Now, it turns out it's a little different than the nicotinic acetylcholine receptor found at the neuromuscular synapse, but nonetheless, it's still a nicotinic acetylcholine receptor. Then the postganglionic neuron to target one of two things. It can either be acetylcholine or it can be norepinephrine. And we'll see that's where we begin to see the distinction between the sympathetic and parasympathetic branches. But whether or not the neurotransmitter is acetylcholine or the neurotransmitter is norepinephrine, the receptor will be a G protein coupled receptor. These G protein coupled receptors we learned about in the endocrine system. Same story. We're going to bind a neurotransmitter to the GPCR. We're going to have this cascade of events, and we'll talk about the cascade of events. When we talk about the autonomic regulation of the heart, the sympathetic branch, the fight or flight branch, tends to increase the heart rate. We'll see it also increases the strength of contraction, but first we're going to talk about the increase in the heart rate. The parasympathetic branch, on the other hand, is the branch that's going to be rest and digest. And so you can imagine the parasympathetic branch is now going to decrease the heart rate. Now, we normally teach these two as sympathetic branch is on and we activate the system, and the parasympathetic branch is on and we inactivate the system. It doesn't really work that way. The way these two systems work is one takes ascendancy and the other falls to the background, but it never turns off entirely. Or then the other may take ascendancy and the other falls to the background. And we're constantly in this balance. And as you're sitting here, quietly sitting there, you're using both your sympathetic and parasympathetic nervous systems to regulate blood pressure, you're using it to regulate your body temperature, you're using it to regulate your heart rate. And that's going on in background all the time, you don't notice it. But it's not as though you turn one system on and you turn the other off, and you turn the other system on and turn the first one off. You don't do that. They're on at all times. It's just which has primacy. So we go through that, think about it. And then finally, we'll end with some of the anatomy. And we'll have established that in both cases, the sympathetic and parasympathetic branch, the preganglionic neuron resides in the central nervous system. Well, we'll see for the sympathetic branch, the cell bodies are located in the spinal cord between T1 and L3. The parasympathetic branch, however, has its preganglionic neural cell bodies in the brainstem. And so we'll see that we're going to now put these in a different place. We'll follow their paths outward. We're going to tell the story, because it's the heart, we're going to tell the story of the vagus nerve. And we were introduced to that when we talked about Dr. Loewy's experiments. But let's start here with that image that we had at the end of the last lecture. And we labeled this as the electrocardiogram. And what the electrocardiogram is measuring is it's measuring the ionic movements in the muscle of the heart. And depending on how many electrodes you use, depending on where you put the electrodes, you'll see different images. But by and large, there are just three phases we want to talk about. And if we draw it this way, this is time, and this is voltage. Typically what we'll see is we'll see this depolarization. And we see this hyperpolarization, up, down, up. And another wave. And as we mentioned yesterday, this is the P wave. This is the QRS complex. And this is the T wave. Well, what do these represent? They represent ion movements into and out of cardiomyocytes. This P wave represents the influx of sodium into atrial cardiomyocytes. This QRS complex represents the influx of sodium into ventricular cardiomyocytes. And finally, the T wave represents the efflux of potassium from ventricular cardiomyocytes. So hold on to this. P wave is atrial contraction. QRS wave is ventricular contraction. T wave is repolarization of the ventricular myocardium. So now let's think about that in terms of the pressure volume curve. When we draw the pressure volume curve again, remember we start out at point A, where this is about 50 mils. This is about 3 to 5 millimeters of mercury. And remember, during this period of diastolic filling, the pressure doesn't change until the very end. We get that little bump, that little bounce. And we're at point B. Remember what that is? Somebody tell me what that is? Atrial contraction. Where's that going to be here? The P wave. So the atrial contraction represents the influx of sodium into those cells that begins the process of excitation-contraction coupling. That reference here is that the P wave is the sodium influx that then leads to the excitation-contraction coupling of the atrium. And then between B and C, this is that period of isovolumetric contraction. Well, when's that going to be? Well, that's the QRS complex. That's the influx of sodium into those ventricular cardiomyocytes. Remember, those cardiomyocytes are contracting and they're generating force. And they're generating more and more force until the aortic valve pops open. And we then enter the ejection phase. So throughout this period, from B to C to D, throughout this period when we're either generating pressure or expelling the blood from the ventricle, throughout that period, we're talking QRS. We're contracting, we're actively contracting that muscle. Then between D and A again, remember this is isovolumetric relaxation. Well, what's happening when that muscle's relaxing? Potassium's coming out. We're repolarizing the membrane. So that means the T wave is going to be during this period of isovolumetric relaxation. And then we have a period where we wait a little bit, and then we have another P wave, QRS complex, another T wave. And so this is the interbeat interval, and what we're talking about here is starting the cycle all over again, but we can see that cycle here in the electrocardiogram, because we're measuring the sodium influx either into atrial cells or into ventricular myocardial cells. Any questions? Yes?
So the A to B, that's the P wave? Or is that...
So A to B. Mm-hmm. So much of this is going to be during this interval between the end of the T wave and the beginning of the next P wave. But the P wave represents the pressure generated by the contraction of the atrial cardiomyocytes. That's where this little bump comes from. That's the pressure. But you're right, trying to impose time on this. During the period of diastolic filling, if we come back to this thing, that's going to be between, let's do it here, that's going to be between the T wave and the next P wave. Right. Anything else? Yes, in the back.
What did you say the isovolumetric relaxation corresponded to on the electrocardiogram?
Oh, the isovolumetric relaxation is the period of time when you're not contracting. So if you're not contracting, potassium will be leaving those cardiomyocytes. So remember, the contraction is associated with sodium influx.
Where is it on the electrocardiogram?
Where is it on the electrocardiogram? It's going to be this T wave. Anything else? Okay. With that, let's move on. And we'll start a discussion about the autonomic nervous system. And in the first step here, I'm going to show it as a 3-box model. This is going to be the generality of the autonomic nervous system. So what we're going to see here in the next few minutes is true for both the sympathetic and the parasympathetic branches. Before we get down into the details of those two branches, I want to use some generalities here. So we can label this as the autonomic nervous system generalities. What I mean by that is we're going to start with a box. And we're going to label this box as the central nervous system. And we'll see that the neuronal cell body for the autonomic nervous system is in the central nervous system, and it sends a process out in the periphery. That links to another box. This box is going to be an autonomic ganglion in the peripheral nervous system. So if you'll remember, the definition of a ganglion is a collection of neuronal cell bodies in the periphery. So out in the periphery, we're going to have these cell bodies, and the preganglionic neuron is going to be represented by that Y. The synapse is going to be represented by the Y onto a postganglionic neuron. So this is the postganglionic neuron. And this cell represents, in blue, represents the preganglionic neuron. And this Y to this dot is meant to be the synapse between the pre and post ganglionic neurons. So the second neuron here in green, I'm going to have to extend this process out. This synapse is in the third box. That we're going to call the target. Now, for the autonomic nervous system, this synapse of the postganglionic neuron onto the preganglionic neuron, this target could be one of three things. It could be heart. It could be heart. This could be either contractile or potentially conductile cells. It could innervate smooth muscle. This could be smooth muscle we'll talk about tomorrow around all of the arteries and arterioles. Smooth muscle of the gut, but it's going to innervate this special kind of muscle called smooth muscle. And the third thing that it potentially could innervate is a gland. Now, our purposes are going to focus on this guy. We're going to focus on the autonomic regulation of the heart. There are a couple of other generalities we can put into this simple graph. We can say that this preganglionic neuron transmitter, this side is always acetylcholine. And we'll abbreviate this as ACH. And always at this site, the receptors. Always at this site, the receptors are nicotinic, acetylcholine, receptors. We abbreviate this usually as small n, ACHR. So this is the receptor, This is the transmitter. Now, to be sure, it's a different nicotinic receptor than we saw with the neuromuscular synapse. But the rules are pretty much the same. It's still an ionotropic channel. It's a fast channel. It binds in the acetylcholine and opens very quickly. So in that sense, it's the same kind of arrangement. And we'll see that here, we release the neurotransmitter, and we set up, using the neurotransmitter acetylcholine, we set up an EPSP. Those EPSPs will summate to fire an action potential to propagate to the output region here. Now, let's leave this here, because now this is where we begin to see the separation of sympathetic and parasympathetic branches. The real distinction between the sympathetic and parasympathetic branches lies in the neurotransmitters and receptors responsible here between the postganglionic neuron and the target. So, any questions up to there? So this is the general term. This is a general morphology. Yes?
Can you repeat the abbreviation that you said, the little n?
Oh, what I'm trying to refer to here, the little n, is the nicotinic. That tells us it's a nicotinic receptor. ACHR tells us the neurotransmitter it's going to bind to. And then we add an R at the end to say that that's the receptor. That's the shorthand. Down here, the abbreviation for the neurotransmitter, acetylcholine, is typically ACH. Okay, let's now begin doing some comparison. Let's now begin thinking about what distinguishes the two branches. So let's spell out some generality and then begin to drill down and see where the differences between the two branches, in fact, lie. Now, to begin with, when you think of the autonomic nervous system, you immediately have to think that there are the two branches, the sympathetic branch and the parasympathetic branch. And although it's a bit of an exaggeration, it usually will work to think of the sympathetic branch as responsible for flight response, and the parasympathetic branch for the rest and digest. So when we talk about the branches, say about the autonomic nervous system branches, we have first the sympathetic branch. We're going to see that for our purposes in the heart, we're going to make the heart run faster. Generally, when we think about the sympathetic branch, we're thinking about fight or flight. Overall, blood pressure goes up, sweating may begin, vessels to the skin become constricted. There are a whole series of things that happen. But what we're going to talk about in the sympathetic branch, the fight or flight idea, is going to be the effects on the heart. And we'll see the sympathetic branch increases heart rate. And as a more subtle effect, it increases ventricular as contractility. That's a fancy term simply to say that the ventricular contraction will be stronger, and we'll see molecularly how that's going to happen. The other branch, the parasympathetic branch, is typically referred to as the rest or digest branch. Now, it's a little bit of an exaggeration, but it kind of works. So when you're standing on the street, car whirls by and you almost get hit by the car, you're going to be in fight or flight. That's sympathetic branch. Your heart rate's going to be faster. You'll see that your pupils will dilate. We do all those things. This is the branch that's most active when you're sitting at home watching TV, eating popcorn. This is the rest and digest branch. This is the one that's most active, we'll see later, in activating the acid secretion from the stomach and working the gut motility and all that sort of thing. But for our purposes here, for the rest and digest branch, we're going to see the effect on the heart is to slow the heart rate. This is a detail, something we'll emphasize, is that where the sympathetic branch actually innervates the ventricular cardiomyocytes and increases the strength of muscle contraction, the parasympathetic branch innervation seems to be almost entirely dedicated to the pacemaker cells. But that's a detail. Parasympathetic branch is going to slow things down. Sympathetic branch is going to speed things up. For our purposes today, that'll work just fine. So those are the two branches. If we come back to this image, it doesn't matter whether we're talking about the sympathetic or parasympathetic branches, the pre to post ganglionic synapse is going to use the neurotransmitter acetylcholine. Where we begin to get into differences is out here at the target. And the difference becomes that the sympathetic neurotransmitter, there are some exceptions, but typically the sympathetic neurotransmitter is norepinephrine. The neurotransmitter for the parasympathetic branch, we'll put this in green. The parasympathetic branch the neurotransmitter is acetylcholine. Now, hold on to this idea, because this is an important idea. Back here we use acetylcholine, fair enough, but in this case it's always a ligand-gated ionotropic channel, fast response. Out here, whether we're talking norepinephrine or we're talking acetylcholine, the receptor is a G protein coupled receptor. We'll see that the acetylcholine is activating a G protein coupled receptor to slow the heart. Norepinephrine is activating a G protein coupled receptor to increase the heart rate. Now we'll work our way into the molecular aspects of that. But if you hold on to the idea, and conceptually you know if you've just been frightened, your heart's beating really fast, so you've increased your heart rate. We're going to increase our heart rate using norepinephrine. If you're sitting there quietly, meditating, you're in parasympathetic drive, your heart rate goes down, you're going to use acetylcholine to reduce the heart rate. So working through a G protein coupled receptor. I'll tell you right now, it's a little bit of foreshadowing, we're going to be working with G protein coupled receptors that are exerting their effects on cyclic AMP. So we can think about this later, but what we're going to see is that the cyclic AMP binding to the HCN channel that's going to regulate increasing or decreasing the heart rate. And we'll see how that gets done. So any questions up there? Yeah.
You keep saying acetylcholine?
Okay, there's a history to this. It's a long history. It's a long history. The history is that when I was a graduate student, my advisor had been trained in England. And so he talked about synapses and vesicles and acetylcholine. And I had been raised to say acetylcholine. But I morphed the two together. And so it became acetylcholine.
Okay.
So that's what it is. Right, it's the same thing. It's the same molecule. That's right, sure. Okay, so let's now think about the anatomy, the autonomics. Okay, now let's start this panel here and talk about the anatomy. Of the autonomic nervous system. We're going to start over here with the sympathetic branch. Now I'm first going to draw this as a cross-section of the spinal cord, and then we're going to superimpose it on the ice cream cone diagram. So if we look at the spinal cord, and we draw the gray matter, we're going to see that between the first thoracic vertebrae called T1, running through the third lumbar vertebra called L3, in the spinal column between T1 and L3, there's a special region of the gray matter, which kind of pooches out here. And this gets a name. This is said to be the intermedio lateral gray column. This is a column of neuronal cell bodies. So in cross-section, you see them just as a bunch of dots. We'll see here in a minute. But if you take this cross-section and put it into the ice cream cone diagram, column of cell bodies. Cell bodies is descend a process down across the ventral horn and out through the ventral root. The path that the axon takes during development is a bit circuitous, so I'm not going to get into that. But from what we have above, you know where we're headed. This is a preganglionic neuron. Cell bodies and central nervous systems in the spinal cord. The process goes out to the periphery, and it's going to form synapses, as we'll use the orange. It's going to form synapses in a site known as the sympathetic chain ganglia. So the sympathetic chain ganglia are going to be a series of ganglia. They used to be known as the paravertebral ganglia. They're right along the spinal column. And there's going to be a pair of these on left and right side between T1 and L3. These are the sites along the spinal column where the preganglionic neuron synapses onto the postganglionic neuron. Now that postganglionic neuron sends a process out, and it's going to innervate the heart. Specifically, it's going to innervate the pacemaker cells. So I'll make this as kind of a green dot and label this as the pacemaker cells. In the SA and AV nodes. Now, the truth of the matter is there'll be branches here, some of them coming to the SA nodes, some to the AV nodes. Some will actually even go to the bundle of His and to the left and right bundle branches. But the long and the short of it, this innervation of these pacemaker cells is going to be the modulation of the heart rate. That's how we're going to modulate the heart rate. This is sympathetic, so this is going to be increasing heart rate. So that's the anatomy of the sympathetic branch. But now let's superimpose that onto our ice cream cone diagram. There's the brainstem, the spinal cord. We're going to start someplace down here of T1 and go to L3. And we have these pairs of spinal nerves, remember? And now if we imagine the bilateral symmetry that we've emphasized and we say this is right, and we say this is right, we can see between T1 and L3 a column of these neuronal cell bodies. These are the neuronal cell bodies which we show here in cross-section as part of the intermediolateral gray column. Since we see this on both sides, we'll see two columns of these cells. These neuronal cell bodies send processes out into the spinal nerve, and they, shortly after this entering the spinal nerve, will form synapses out here in the sympathetic chain ganglia. It's not perfect, but pretty much what we see here is that we have, for each segment, we have a ganglion. From what we know in the upper panel, we know that this preganglionic, this is labeled, this is the preganglionic sympathetic neuron is going to synapse onto the postganglionic neuron. And this, of course, is going to send its process out to the heart, just as we showed here. Now, I should say, because we're bilaterally symmetrical, you'll see a similar set coming from the left side, coming out into the spinal nerve, forming synapses in the sympathetic chain ganglion. And the postganglionic neuron now innervating the heart again. To emphasize the point, the neurotransmitter here, neurotransmitter is acetylcholine, and the receptor is the nicotinic acetylcholine receptor. The neurotransmitter out here is going to be norepinephrine. You may sometimes see this referred to as noradrenaline. It's the same thing. As adrenaline, norepinephrine is the same thing as noradrenaline. The same molecules, just different names. The norepinephrine is now going to be the neurotransmitter released by the postganglionic sympathetic neuron. So that's basically the outline of the sympathetic branch. Any questions on that? Well, let's now then go back up to the upper board, and we'll pretty much do the same thing. But now we're going to talk about the parasympathetics. And one of the things to hold on to here is the difference in this CNS location. For the sympathetic branch, the CNS location is always the spinal cord. Always, always, always. For the parasympathetic branch, the central nervous system location is always the brainstem. Now, if you take a neuroanatomy course, you'll find that there are a number of potential sites in the brain where we can have these parasympathetic neurons. And those preganglionic parasympathetic neurons may send processes out into cranial nerves 3, 7, and 9. But in fact, and 10. The only one we're 3, 7, and 10. I'm sorry. I don't think there's 9 in there. But the ones we're going to worry about is 10. And so let's take this panel and describe the parasympathetic branch. Anatomy. Now, since we're focusing on the heart, we're going to tell the story from the point of view of what's going on in the brainstem. So this is the brainstem, just as we have above. Here's the spinal cord. But now, in the brainstem, there's a cluster of neurons called the motor nucleus of cranial Well, cranial nerve 10 is another name. The other name, I really like this name for cranial nerve 10. Usually I prefer the numbers, but in this case it's called the vagus. And it gets its name vagus because it comes from the Latin meaning the wanderer. And it's a great name because these neuronal cell bodies up here in the brainstem, the motor nucleus of the vagus, or the 10, send processes out into this cranial nerve. This cranial nerve is the vagus nerve. It's cranial nerve 10. And these preganglionic parasympathetic neurons, the processes, the conductor regions of the preganglionic parasympathetic neurons go out into the periphery in the vagus nerve, and they go throughout the thoracic and abdominal cavities. So all of the parasympathetic innervation of the thoracic and abdominal cavities, there's some question about the genitalia, that may be in the sacral spinal cord, but we're talking about the vagus going all over the abdominal and thoracic cavities and innervating targets in that region. So the vagus is a great nerve. It's wandering all over the place. It's bilaterally symmetrical, so it comes out both sides. But the one we're interested in is we're interested in the neurons that are headed to a ganglion. And this is going to be the cardiac ganglion. So remember, we've got to obey the rules. Preganglionic neuron cell body is in the central nervous system. We're in the brainstem. Sends a process into the periphery. The rule is it has to have the preganglionic neuron synapse onto a postganglionic neuron in a ganglion. This is going to be in the cardiac ganglion for the heart. The cardiac ganglion is going to be located in the wall of the right atrium. Now, if we were to follow one of these other neurons whose cell body is in the motor nucleus of 10, and we were taking it to, say, the liver or someplace, the ganglion would be in the wall of that organ. So here's one of those other interesting contrasts. When we're talking about the sympathetic branch, the pre to postganglionic synapse is in the sympathetic chain ganglia, right along the spinal cord for the most part. Whereas in this case, there's great long neurons extend from the brainstem out all the way to the organ that will be innervated, but there's not direct innervation. There's an interlocutor here. And the interlocutor is going to be, let's use the green one. The interlocutor is going to be the postganglionic parasympathetic neurons. And if we look closely at one of these cardiac ganglia, if we try to put it into context, if we use the simple heart, where this is going to be the right side, we're going to see that the ganglion will receive synaptic inputs from this preganglionic parasympathetic neuron. But then the postganglionic neuron, which we're going to put in green, this has the cell bodies in this cardiac ganglion in red, but it sends out fairly short processes. And these will now innervate the postganglionic parasympathetic neurons, innervate pacemaker cells. Last couple of minutes, let me go to the back panel. I don't think I'm going to get through both of these, so I'm going to start here with the sympathetic branch. Now I'm going to label this as innervation of pacemaker cells. By postganglionic sympathetic neurons. And I'm going to take this pacemaker cell and I'm going to show it in green because that's the form I used yesterday. We're going to imagine that there's this green neuron up here, this sympathetic neuron. And it's releasing neurotransmitter. Here the neurotransmitter in this case is norepinephrine. The receptor here, the receptor I'll put in red, is a GPCR. Particularly, it's a GPCR that's said to be a muscarinic. Acetylcholine receptor. As a detail, the particular muscarinic type here is M2. The story is, and we'll start here and then we'll finish it tomorrow, is that receptor binds, sorry. The GPCR in this case is the norepinephrine receptor. So in this case, we're releasing norepinephrine. We have a norepinephrine receptor. It's a GPCR. And what we're going to see tomorrow is it increases the activity of adenylyl cyclase. Remember what adenylyl cyclase does. It's an enzyme that generates cyclic AMP. So what we'll do tomorrow is we'll bring around the idea of generating the cyclic AMP and that HCN channel. And to remember that the cyclic AMP must bind the HCN channel to open those sodium channels to let sodium in. And that's how you're going to increase the heart rate using the sympathetic branch of the autonomic nervous system. With that, I'll let you go.